I built a modules combined in a planned out way. Each new piece must be told where to go.
Welcome back to AP Bio Immune System Part 2. And we're going to pick up where we left off and discuss how those phagocytes that engulf and digest an antigen use that antigen to identify what kind of antigen it really is and how the immune system determines what to do next, how to mount a response. So what we're going to do is we're going to focus for a moment on a wound. And in this case, we have a problem that occurred to the skin. And you notice this wound is an abrasion or a cut, and bacteria are our pathogens. Pathogens cause disease, cause problems to the body's health and homeostasis, and they contain antigens, foreign proteins that are non-self. And so immediately what happens is mast cells are alerted to this entrance of bacteria, of antigens into the body. The first line of defense has been breached. And so what happens is all of these mast cells, which lie underneath the surface of the skin, begin to release histamines and prostaglandins. And what the histamines do is they cause this local swelling to occur. And that local swelling is when these blood vessels, and here we have a capillary, for example, this capillary begins to dilate. And when it dilates, what that means is it gets wider. And when it widens, it allows more blood to flow to the area. Prostaglandins, another hormone that's released, a protein locally, a local signal, allows more blood flow to come there, more white blood cells to get there, and makes the area hot so that those antigens are uncomfortable and their proteins begin to denature. So you get a local fever. Again, imagine getting a splinter and having that splinter, it throbs and it gets hot, and it's painful and it's swollen until you pull that piece of wood out of your skin. Well, you've pushed bacteria in there as well. And so those bacteria have to be dealt with. And so what happens is this white blood cell here is traveling through the capillary, and it's able to slip through an opening that you see in the wall of the endothelial lining of the capillary. The capillary is made of a thin layer of cells, and those cells open a little bit and make a pore, and that white blood cell is able to slip out of that pore in a process called diapedesis. And in diapedesis, that's when the white blood cell leaks out of your bloodstream and into the spaces between your cells. And in this case, that white blood cell is a neutrophil. And what that neutrophil does is it starts engulfing all of those bacteria that start entering. And now there's many neutrophils that enter. And so many neutrophils enter and begin to die from eating these bacteria that they begin to form pus when the neutrophils die by eating up the bacteria. It makes just a big mess in there. But that mess is important because it starts the healing process. Now what also happens is not all of the phagocytes are neutrophils. Some of those phagocytes, let's try that again, some of those phagocytes are macrophages. Macrophages are the large phagocytes, and their job is to do two things. Grab the bacteria, which it's doing here, and put it in a vacuole, and also release cytokines. Cytokines are chemical messengers called interleukins. And what they do is they are local signaling molecules. And those signaling molecules alert the other white blood cells, especially helper T cells, something is up. Start the healing process, but also start the identification process of what kind of antigens have entered the body. Here we have a video that shows white blood cells, these large cells, doing phagocytosis on the tiny cells, the bacteria that you see in the picture. And you're going to notice some of these cells are starting to do phagocytosis and bringing those cells into vacuoles. And as the vacuoles engulf the bacteria, lysosomes break down all of those bacteria into tiny little components. Those little components are the proteins on the bacteria's membranes that are then used by the white blood cells to identify what kind of pathogens they are. And so these white blood cells are large monocytes or large phagocytes. They're these guys here. They have to start talking to the T cells. These are our lymphocytes. Lymphocytes are so called because they spend a lot of time in the lymph. And so the phagocytes are going to present the antigens to the T cells, especially the T helper cells. Now the neutrophils, if you remember, those are all those little cells that form pus. They make up about 40 to 70% of all of your white blood cells. They're kind of like on the front lines. All of the other cells are much smaller in number. Eosinophils make up maybe a maximum of 6% or so, and then basophils are less than 1%. The numbers of monocytes and lymphocytes vary based on what the level of infection is that you have. And so that's kind of important to pay attention to. A high white blood cell count indicates there's an infection. So here we have a picture of an antigen-presenting cell. This is our phagocyte. This is the cell that just engulfed one of those bacteria. And now what it does is after it digests that bacteria, so I've got a bacteria sitting in this vacuole here, and this little vacuole begins to fuse with the lysosome that's here, and it digests and breaks down this bacteria into tiny little pieces. The pieces of bacteria become the antigen that this phagocyte is showing to our T helper cell. And the T helper cell is the one that starts with our specific immune response. Because what does it do? Well, it has to determine self versus non-self and what level of response does it make. The antigen presenting cell is very non-specific in its response. Remember, its job is just to gobble stuff up. Once it's gobbled it up and it presents the antigen to the T helper cell, that cell then makes the decision of how specific a response to make. Now, in order to do it, this T helper cell uses what are called CD4 receptors. And these CD4 receptors read this particular antigen. So the antigen is read by CD4 receptors on that helper T cell. This receptor here, the hand that is holding that antigen, is called an MHC receptor, the major histocompatibility complex receptor. It is what helps determine self from non-self. So in order for this whole process to work, you have to have MHC receptors and CD4 receptors work together to identify self from non-self. This is that local signaling we've been talking about. Very, very close, like a handshake. Literally like a handshake. So to determine self from non-self, we use what's called the major histocompatibility complex. The word histo means tissue. And the word compatibility means, well, how much of a match there is, how compatible. So is there a tissue match or is it non-self? And so in order to determine non-self from self, the cells, the helper T cells, read the cell surface glycoproteins. So a macrophage will present an antigen to a T helper cell, and the T cell determines if it's non-self. And if it is, it leads to an immune response. These little proteins sitting on the surface of this T helper cell are called CD4 receptors, and their job is to determine self from non-self. So here we have this situation again. Antigen presenting cell is right here. This is our antigen presenting cell. This is our phagocyte. The phagocyte is eating our antigen. This is our antigen. And if you notice, our poor little antigen is getting digested. And a little tiny piece of it is sitting on the surface of the cell membrane of our antigen presenting cell. Our antigen presenting cell shows the antigen to the helper T cell, and it uses the CD4 receptors to determine self versus non-self. This is, again, the major histocompatibility complex. How compatible are our tissues? Now, if we look more carefully at the picture on the right, here's our T cell in purple, just like over here. Here's our macrophage in teal, just like over here on the left side. And look at the names of these proteins. Here's the CD4 receptor. That's on the T helper cell. It is reading the antigen, which is right there, that little red thing. That's the antigen. That's a piece of the bacteria. It is being presented to the T cell by our antigen presenting cell. Our antigen presenting cell is using a protein called MHC, which is major histocompatibility complex protein, holding the antigen out for the T cell to read. This is a handshake. This is very, very close local signaling. Very important because it allows the cells to touch and communicate with each other. Very important information. So what happens next? Well, the T helper cell releases a whole bunch of cytokines, again, these little proteins called interleukins. And what they do is those proteins activate the T cytotoxic cells. In other words, the killer T cells called cytotoxics. These cells are like ninjas, and their job is to go around stabbing cells that are infected. And so look over here on the left. This is a killer T cell, a cytotoxic. It literally blows up virus-infected cells, your own cells, cancer cells that have gone rogue, and bacterium-infected cells of your own body. So the way this happens, if we look down here, this is our cytotoxic T cell. It has a CD8 protein, very similar, to identify self versus non-self. There's our MHC protein on the infected cell. This infected cell is one of your body cells. And look, it's holding out little pieces of antigen. This is holding out little parts of the antigens from the bacteria saying, I shouldn't be here, I'm infected, please kill me. And so this poor cell is screaming out, kill me. And so what does this cytotoxic T cell do? Well, it starts to release a chemical called perforin. Perforin are these little orange or whatever color dots these are, red dots here. These are released from vesicles inside of the cytotoxic T cell, and they punch holes inside of an infected cell, so that at the end of this process, this cell pops. It explodes and dies by apoptosis. It's so amazing. Literally, your immune cells are killing your infected cells because your infected cells are advertising. Look, they're infected. They're presenting little infectious particles. And so in comes the ninja cells, the cytotoxics, to destroy them. This is a wonderful image of a cancer cell being attacked by a cytotoxic T cell, which is popping lethal holes by releasing into that cell, let's try that, all of these chemicals called perforins. I'll try to spell this correctly. Perforins. Perforins are proteins that pop holes in infected cells. And what do they cause? Well, they cause the cell to do apoptosis. Pop them, blow them up. So to summarize, this is, first of all, this is a adaptive immunity. Called cell-mediated or cell-to-cell immunity. And what this means is that your cells are literally walking up to another cell and saying, hey, are you infected? And if your body cell is saying, yeah, I'm infected, well, your cytotoxics are going to pop it. They're going to kill it. They're going to say, okay, I have to slice you open with perforin. I'm sorry, you're dead. And that's for the good of all the other cells around it. This is adaptive immunity because your cells have to learn what is the pathogen, the antigen that I'm looking for. It's not innate. It's not like skin or mucus that you just make automatically and it blocks everything. This is specific. This is adaptive. You learn over time to distinguish all the different pathogens and antigens out there. There's 3 cells that do this. You have the helper T cells, which are like the generals. They use CD4 proteins to bind to major histocompatibility proteins to determine, hey, you're self or non-self. If it's non-self, the helper T cells then activate cytotoxic T cells. Cytotoxic T cells are the ninjas. They have CD8 proteins that bind to major histocompatibility proteins on the infected cells, tumor cells, cancer cells. Their job is to be lethal, to pop cells that are infected. This is a ninja. Hand-to-hand combat, up close and personal. In order to prevent the ninjas from going overboard and just wiping everything off the map in your body and killing you, you have regulatory cells, suppressor cells, cells that say, whoa, slow down, we're only killing pathogens and antigens. This is not an autoimmune disease. We don't want to start destroying the pancreas, like in diabetes type 1, or the nerve cell Schwann cells, the myelin sheath, like in multiple sclerosis. Those are autoimmune disorders where you have your white blood cells out of control and attacking your own body. Regulatory cells say to the cytotoxics, stop. Whereas helper T cells say to the cytotoxics, go. And so there's a delicate balance there. Notice that these regulatory cells have CD4 and CD25. These are cell-determining proteins. That's what CD4 means. Cell-differentiating or cell-determining proteins. Okay. So finally, let's talk about humoral immunity. Humoral immunity is another kind of adaptive immunity that involves your B cells. And the word humoral means the fluids. If the cytotoxic T cells are ninjas, then the B cells are like snipers. They kill from long distances. And the B cells use very special bullets called antibodies or immunoglobulins. And these immunoglobulins, or IGs, are Y-shaped proteins. They are single-use proteins. They make tremendous amounts of them, they shoot them and use them to stop antigens. And so when we look at the picture here, step 1 is showing a naive B cell learning about which bacteria are pathogenic are antigens, don't belong. These are not your microbiome bacteria. These are the bad guys. The helper T cell also helps to teach the B cell by releasing interleukins that say, hey, wake up. Wake up and start producing more cells. Do mitosis. So the B cell does mitosis. It makes plasma cells, which are involved in your primary immune response. Primary immune response means it takes about 7 to 10 days to make your antibodies to fight a disease. And it also makes memory cells. That's part of your secondary immune response. And your secondary immune response is faster, 3 to 4 days to produce antibodies. These memory cells remember every disease you've ever been exposed to so that you can mount a quick response the second time you're infected. So let's pay attention to the plasma cells right now. Here's our plasma cell in step 4. Look what this plasma cell is doing. It's coughing out antibodies. It's shooting antibodies at a distance. These are immunoglobulins, our Igs, at a distance, and they're attaching to our bacteria's little antigens, these little proteins, immobilizing them and marking them for destruction by a phagocyte. Now the phagocyte comes in, phagocyte goes nom nom nom, eats it up, gets rid of the bacteria. The memory cell, of course, will remember this whole thing, and the second time you're infected by the same bacteria or the same virus, again, in the future, your response is faster. A quick note about how your antigens actually work. How your antibodies, actually work against antigens. There are probably 10,000 different antigens out there in the world. There are probably, I think there are exactly 5 different immunoglobulins. Again, those are your Y-shaped proteins. So how do you take 5 different kinds of antibodies and fight against 10,000 different antigens? Well, the answer has to do with the shape of the Y. If you notice up here, we have what's known as the variable region of our antibody. Remember, antibodies are good. This is what your B cells are making. So this is a B cell right here. This is the cell membrane. And so that variable region can change its shape to match any one of the 10,000 antigens. How? Well, look over here on the right. Genes control the production of proteins. We know this. Transcription is when DNA makes RNA, and translation is when RNA makes the proteins at the ribosomes. Well, what you are is adaptable. You can adapt to what antigens are attacking you by removing sections of DNA that you don't need and isolating that DNA from your chromosome and only producing the kinds of antibodies that you do need. So maybe this set of genes here produces the shape that you see here to fight only this antigen there. But a different combination of genes, different gene combinations, create different antibody shapes. And this is what protects us from all those different antigens out there. This is what we mean by adaptive immunity. You adapt your antibodies to shape the antigens that are coming in, to match the shape of those antigens coming in, which is amazing. So here we have the process kind of summarized for B cells. Imagine somewhere over here, I'm going to make a little B cell, a little plasma cell, and this little plasma cell is shooting out all of these little antibodies. Pew, pew, right? Shoots them out into the blood. And so here we have all of these circulating antigens. Okay? The antigens are in red. The antibodies are in blue. These are the good guys. These are the bad guys. And so there are three processes that can happen. The first thing we can have is something called opsonization. Opsonization is when we mark the antigen for destruction. It's sort of like lighting it up with a laser so a smart bomb can come in and destroy it. And so these antibodies here are marking this antigen to be destroyed. What's going to destroy it? Well, a white blood cell is going to come in and it's going to do phagocytosis. Nom, nom, nom. Nom, nom, nom. Okay, eat it up. The second thing that can happen here in B is called neutralization. Neutralization, I think that's spelled with an S. Oh, let's try that again. Neutralization is when you prevent the antigen from binding or attaching to a body cell. And so if you notice, all of these antibodies here, they're surrounding that antigen. It can't do anything. It can't go anywhere. It's trapped. Final process in 2C, this thing here, you see, you got a whole bunch of antigens attached to a whole bunch of antibodies. That's called agglutination. And agglutination is another fancy word for clumping. And so when we clump all this crap together, what ends up happening is a white blood cell is going to come in, a giant phagocyte, and it's going to gobble all of that up and go nom, nom, nom. Actually, you can see that happening at the bottom here. So you see in step 3, the bottom, here is our, this one looks like it's opsonization going on here. And we have the phagocyte in step 4 is gobbling up and putting all of that into a vacuole, right? And so here, that's terrible. Let's try this again. Here, we have a vacuole that's forming. And that vacuole is keeping the antibody with the antigen trapped, and it's going to get digested and destroyed. Remember, antibodies are like bullets. Once they're used, you can't usually reuse them. There are 5 different kinds of immunoglobulins, and so IG stands for immunoglobulin. They are IgG, IgA, IgM, IgE, and IgD. Gamed. And so these are the 5 that I was talking about. Remember, they have the variable region right here. And so this can change. This is their variable region. This is coded for by genes, which allow different shape of variable regions to match the different shapes of antigens. The one that you really need to know is IgGs. 80% of all antibodies are IgGs, and they are antiviral. And so, if we're trying to produce some sort of passive immunity to stop the COVID-19, and you want to transfer somebody's antibodies who's recovered from COVID-19, you can take those antibodies, the IgGs, that match the coronavirus, and you can transfuse the plasma that contains those antibodies into a person who's sick, and that will help boost their immune system. These are what you would actually transfer, the IgGs. The other ones here make up the other 20%, and most of these are found either in breast milk or in mucous membranes. They make up a smaller percentage. Some of them are for allergies, some of them are for parasites and bacteria, etc. They all have slightly different shapes to them, but the IgGs are probably the most important. Really quickly, here's a graph that shows us primary. Oh, for the love of God. Let's try that again in yellow. Primary response versus a secondary immune response. Primary immune response takes 7 days to build up antibodies from a naive B cell to an activated B cell to a B cell that's making a tremendous amount of IgGs to stop an antigen. And here is our antigen. It takes 7 days to build up enough IgGs. However, because you make some memory cells, if you are infected by the same antigen again, it only takes 3 days for a secondary immune response because you don't have naive B cells anymore. You have memory B cells, and they can produce IgGs a lot faster with a bigger response. Look at the peak here. This peak is much higher than the peak response over here. And so a secondary response is much more aggressive to fight off an antigen than the primary response was. These pictures appear in your packet. You can review those for help. You're going to read an article about immunotherapy and the coronavirus from the New York Times. Please make sure you take a look at that. And there's some summary facts that you're going to include in Microsoft Teams for this week's assignment. Finally, a word about HIV. HIV is a problem because it's a RNA virus, but remember it's a retrovirus. A retrovirus uses a special enzyme called reverse transcriptase. And so what this virus does, take a look at these receptor proteins, these little glycoproteins sitting in here. What are they going to do? Well, let's take a look. These little proteins attach to your CD4 receptors of a helper T cell, the most important cell of your immune system. Because what happens is HIV knocks out your helper T cell's ability to warn your immune system of the differences between self and non-self. Instead, your helper T cells are infected by HIV's RNA, which converts, using reverse transcriptase, the RNA to DNA. It puts its DNA into your DNA, and then your ribosomes down here, that's a ribosome. Look what your ribosome is doing. Your ribosome is making more viruses. So what does this do over time? Over time, it knocks out your immune system. And so what happens is HIV causes your T cell count to drop. Then they try to mount to come back, and then they're like, nope, we're all getting infected by HIV, and your T cell count drops, well, really low. At the same time, the viral load increases, then it decreases for a little bit because your T cell count goes up in order to mount a response, but it doesn't last. And then the viral load increases to the point where you will die. That could now take decades because of medications and PrEP, which helps to stop the virus's enzymes from integrating its DNA into our DNA, but still, HIV is a problem. We normally have a white blood cell T cell count of about 1,000 cells per cubic millimeter. These T helper cells are called CD4 cells. When your number gets down around 200 cells per cubic millimeter, you have AIDS. And at that point, you need to get checked every 3 months or so for your T cell count, go on medications to slow the spread of the virus, and also take precautions when with being with somebody else. And make sure that you wear protection and inform them that you are HIV positive. I think that concludes the second part of our lesson today. I'm going to entertain any questions you may have on Teams or through Remind, or email me. I look forward to hearing from you. I hope you enjoyed this lesson. Have a great day. Looking at the logic in the ways that we grow. Every gene directed by a signal key code. Proteins that can activate, enhance, or veto. Evo diva. The signals are controlled by other genes that signal. Calculating in a network labyrinth, though, where the heart and liver and the hands and feet go. Signal, 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 signal. Signal mapping to this region. What it ought to be.